Thank you very much for that, and good morning, everyone. Afternoon. I hope you can hear me all right. Thanks so much for that brief introduction, and yeah, it's my absolute pleasure today to be able to introduce two of our brilliant PhD students, Lizo and Nina. I'll introduce them both a little bit more, but then, as Paul's already said, we'll have the combined Q&A at the end. So both Nina and Lizo are funded through the CENTA DTP, so that brings together a number of universities and institutions across the Midlands, as well as external partners like ourselves, and it's very much focused on research and knowledge in earth and environmental sciences. And it's one of a number of these DTP groups that is really hoping to build on collaborative research, taking the best kind of both worlds, really, of being within universities and in institutions like Kew, where we have really groundbreaking research going on. So Lizo's research, you've already heard a bit about the title, but he's supervised here by Bat Vorontsova and at the University of Leicester by Professor Pat Heslop-Harrison. And Nina is split between Kew and the Department of Chemistry at the University of Warwick, where she's supervised by Dr. Tom Whale and Professor Matt Gibson. I should say here at Kew then she's been under Danny Ballesteros. So without further ado, I shall hand over to Lizo to kick us off. Lizo, are you ready?
Thank you, Lisa. Yeah, I'm ready.
Fab, over to you.
Can you see my screen? Hello?
I can, yeah.
Okay. All right. So yeah, thank you, Lisa. Let me just start the presentation. So yeah, I'm Lizo Masters, and again, today I'll be talking about the work I've been doing in the PhD funded by CENTA, looking at the diversity of Urochloa grasses and their application in sustainable tropical forage systems. So Urochloa is a fairly large genus. It currently contains around 130 species, and they're found around the tropical and subtropical regions of the world, although the majority of these species are endemic to or naturally occur on the African continent. Part of why it's such a large genus is that it currently contains species that used to be placed in Brachiaria and Urochloa, as well as a few species from Megatharsis and Scutacni. So as a result, Urochloa is a very morphologically diverse genus. And from an economic point of view, it's a very important genus because a handful of species are used to feed livestock around tropical countries around the globe. So this slide here is just demonstrating the wide array of morphological variability you can see in the inflorescences of Urochloa grasses. So just these 6 species I've pictured are quite different from each other, and that's just the tip of the iceberg for that morphological diversity. From an agricultural point of view, Urochloa forage systems are dominated by 2 agamic asexual reproductive species complexes. The first complex being the Brasantha complex, containing Urochloa decumbens, Ruziziensis, and Brasantha, and the second one being the Humidicola complex, containing Urochloa humidicola and Dictyonura. Now, both of these species complexes are endemic to sub-Saharan Africa, but they're not very widely used as forage crops to feed livestock on the African continent. Instead, they're especially popular in South American countries like Colombia and Brazil. Now, within respective species complexes, there's a wide amount of morphological overlap between respective species. There's a great deal of hybridization taking place between species in respective complexes, and there's also rampant polyploidy within both complexes as well. And that really obscures species limits within these agriculturally useful Urochloas. And then that in turn often hinders breeders' abilities to sustainably produce, more sustainable Urochloa cultivars. So to that end, we looked to use a phylogenomic method to infer relations and species limits within Urochloa sensillato. And we also hope to explore the evolution of Urochloa forage grasses within the 2 species complexes and identify crop wild relatives for those species. So we currently have 142 samples. This represents roughly a third of the total Urochloa species at present and a couple of outgroups. So 59 of these samples were donated by CIAT in Colombia and the USDA to researchers at the University of Leicester and the Earlham Institute. These 59 samples predominantly comprise of specimens within the two agriculturally useful species complexes. And researchers at the two UK institutions then performed RNA sequencing to give us the 59 transcriptomes that were used for our analysis. A further 40 specimens were taken from Kew's herbarium. These were sampled by various researchers in the PAFTL team, my supervisor Alex Zentini and Jan Heckel, who was kind enough to give me some of the samples. And then a further 45 samples of various Urochloa species were collected by me, also taken from the Kew herbarium, where I also did the lab work for the sequencing. And so finally, we used the angiosperm 353 loci set to infer a species tree for Urochloa broadly. So you can see here, this is the species tree that we obtained, and I've highlighted in yellow and blue two clades. One clade contains Urochloa humidicola and dictyonera, and you can see that these species are embedded within a clade that contains some wild relatives to this species complex. And then the second clade contains Urochloa decumbens and resiziensis, and you can see that this reflects how closely related these two species are. Now here at the bottom, I've just enlarged a node that contains all of our samples for Urochloa brasantha. And you'll notice that brasantha is actually split into two fairly well-supported groups. The first group only contains Urochloa brasanthas, which were collected from Kew's herbarium. The second clade only contains brasanthas that were donated by CIAT in Colombia for RNA sequencing. And so we were quite surprised that we retrieved this topology for Urochloa brasantha. We didn't expect such a strong split between the two areas where we obtained our samples, and we were really stumped as to why we were getting this topology. And given what we know about Urochloa brasantha, it's as mentioned before, a polyploid species with various ploidy levels across individuals in the species. And it also frequently hybridizes both in the wild and in the agricultural sphere. And it is likely of hybrid origin as well. And so it's likely the case that this tree-based method that we've used is not really reflecting the amount of genomic variability that is actually present in Urochloa brasantha. And that's not just true for Urochloa brasantha, it's likely true for a number of Urochloa species in our tree. So to help untap some of this genomic variability that's being hidden, we decided to use the bioinformatic workflow HYBE phaser. HYBE phaser is designed to tease apart or to phase gene variants within target enrichment datasets. And this is then used to detect potential hybrids within your specimens, and also to detect the potential parental lineages that gave rise to these hybrid species. So there are a lot of benefits to using HYBE phaser. Firstly, the phasing process takes place on the sequence reads and not on the sequences themselves. HYBE phaser is also built on or built upon outputs from HYBE Piper. And connected to that, as I mentioned earlier, HYBE phaser is specifically designed to be used in target enrichment data sets like the angiosperm 353 loci sets that we used in Urochloa. So the first step in the HYBE phaser pipeline is to take the assembled sequences and the on-target reads for those sequences produced by HYBE Piper. And then using your on-target reads, HYBE phaser will assess the number and distribution of single nucleotide polymorphisms, or SNPs, for each loci in your samples. And then once it's collated that data, what it will do is it will take each of the sequences and it will recode sites where single nucleotide polymorphisms or SNPs are present with an ambiguity code. And basically what that means is you get a consensus sequence for all of your genes for all of your samples, and you can use these consensus sequences for phylogenetic analysis further downstream. So in addition to these consensus sequences, HYBE phaser also produces statistics on loci heterozygosity and allele diversity. So loci heterozygosity simply means the percentage of loci that contain SNPs per sample. And allele diversity simply means the percentage of SNPs across all of your loci per sample, which basically means for all of the base pairs you sequenced for a given sample, what percentage of those base pairs contain SNPs. And now theoretically, what should happen is any species or any taxa that is of recent enough hybrid origin should have a high score for loci heterozygosity and for allele diversity. And the reason for this is a species of recent hybrid origin will have inherited alleles from two parental lineages that are or should be relatively divergent from one another, resulting in the inheritance of divergent alleles in a single species. So once we had gotten our consensus sequences, I then re-inferred a species tree for all of our samples. I then plotted alongside the tips of that species tree the loci heterozygosity and allele divergence for each sample in our tree. And the reason I did this is because the next step in Hive Phaser is to choose a clade reference. Now, a clade reference is essentially a taxa or a species that is not of hybrid origin, so it will have low loci heterozygosity and low allele divergence. And because these taxa that you've chosen as clade references will pretty much represent a clade or a lineage that is not itself of recent hybrid origin, but could potentially be the parent or the parental lineage of a species within your tree that is a hybrid. So effectively what we did is, or what I did is I chose 20 taxa to act as clade references. And then the next step for Hive Phaser is to take all of the on-target reads for all of your samples in your tree and map them to all of your clade references. And what you should find is for species that are not hybrids, the large majority or the majority of reads should all be mapped to a single clade, namely the clade that that species is from. But if your species is a hybrid, then it's very likely that you'll see a large proportion of reads mapping to two or even more different clades, these clades being the parental clades or the parental lineages that gave rise to the hybrid species. So when we apply that to Urochloa, what I've got here is just a zoomed in node that contains all of the Brasantha specimens in our data set. And then mapped next to it is a heat map showing all of the on-target reads that unambiguously mapped to all of the clade references. So for example, in the first column of the heat map, you can see is very dark. So a large proportion of reads for all of the Brasantha specimens matched to the Brasantha clade reference, which makes sense because they're all Brasanthas. But if you follow your eye to where the arrow is pointing, you'll see something quite interesting is happening. For all of the Brasantha specimens from CIAT's agricultural fields in Colombia, a decent proportion of reads are mapping to this clade called the Fusca clade, where you can see with all of the Brasanthas from Kew's herbarium, virtually no reads are mapping to this clade here. And alongside, if you look at the two clades right next to Fusca, you have Urochloa oligobrachiata and lindiensis. You can see a decent proportion of reads mapping to these two clade references, whereas comparatively very few reads are mapping to these two clade references in Brasanthas from CIAT. Now, if we look at the Humidicola dictyonura species complex, we see an identical pattern emerging. We can see that a good amount of on-target reads are mapping to the Urochloa fusca clade for Humidicolas coming from CIAT in Colombia, whereas our herbarium Humidicolas and dictyonuras barely have any mapped reads falling into the fusca clade. And then you can see for Oligobrachiata and lindiensis, a small but noticeable proportion of reads are mapping to these two clades in our herbarium material, whereas virtually nothing is mapping to these two clades for CIAT accessions. Now, when we dive into what these three clades actually are, Urochloa oligobrachiata and lindiensis are two wild African Urochloa grasses. So it makes sense that you'd see some proportion of reads from Urochloa brasantha, Humidicola, and dictyonura mapping to these two African clades because they are also of African origin, and these are potential wild relatives to these agriculturally used species. But Urochloa fusca is not an African Urochloa grass. Instead, Urochloa fusca is a wild grass that's endemic to the Americas. It can be found from the southern United States all the way to central South America. Not much is known about Urochloa fusca, but we do know that it's a diploid species and that early molecular phylogenetic analysis shows that it's closely related to Urochloa arizonica and U. mollis. These two species are also wild American Urochloa grasses. So the question then becomes, why would it be the case that you would see reeds from Urochloa brasantha and Humidicoa complex species used in CIAT? Why would you see these reeds mapping to the fusca clade? And our initial hypothesis at the moment is that either Urochloa fusca itself or a close relative of Urochloa fusca somewhere in South America, in Cali and Colombia, these wild South American Urochloa grasses are introgressing with Urochloa brasantha and Humidicola complex species that are growing in agricultural fields at CIAT. And this is a pretty interesting finding, and we hope to explore it further. And we feel that this finding might go some way to explain why our initial species tree had such a strong split between CIAT brasantha specimens and specimens from Kew's herbarium, which would have been taken directly from African countries. And so going forward, what I hope to do in the next couple of months is to complete the hide phase or workflow. I still have to actually phase hybrid samples so that we can separate gene variants and then use these new phased sequences to either infer another phylogenomic tree or pick a handful of our specimens to do a phylogenomic network. We'd also, as I said, like to explore further these potential introgression events that are happening between Urochloa fusca clade species and the agriculturally important Urochloa species complexes. And then finally, we hope to infer a large chloroplast phylogeny for Urochloa sensillata so that we can supplement our nuclear data set with chloroplast data. And yeah, that's all from my side. Thank you.